Starting now.
Hello everyone, my name is Jonas Korlach. I'm Chief Scientific Officer at Pacific Biosciences, and I'd like to thank the organizers for giving me an opportunity to speak to you today with regard to CRISPR-Cas9 related topics. My talk has two parts. First, I'd like to talk about the importance, the challenges, and new solutions to measuring outcomes of CRISPR-Cas9 experiments. And in the second part of my talk, I would like to mention new methods that can leverage the specificity of the Cas9 enzyme for targeted sequencing of previously very difficult regions. So for the first part, as an introduction, I think you all know that CRISPR experiments are very easy to carry out, and I've just highlighted two examples here in the popular press. Gene editing is so easy to do that we couldn't stop if we wanted to, and there are even these homebrew mail order CRISPR kits and so forth, so allowing absolutely anyone to hack DNA. And so that just is and there are many research papers, obviously, and commercial kits now that highlight how simple and how straightforward it is to do these types of experiments and edit the genome of different organisms. However, it needs to be recognized that it's much, much harder to measure accurately and comprehensively what actually happens when you do such an experiment. And, there's one desired outcome, which is the editing of the target region, but because the CRISPR-Cas9 enzyme system introduces double-strand breaks in the DNA, it has been recognized that there are potential undesirable outcomes, and they are categorized at occurring either at or near the target region, the region that people are interested in editing, and I'll refer to those as on-target regions. And then it has also been described that undesirable events, outcomes can happen far away from the target region, and I'll refer those to off-target events. And so this was described a couple of years ago. On the right, you see a nice article by the scientists, New Methods to Detect Off-Target Mutations, where the article describes bioinformatic methods, software methods to predict similarities of the guide RNA and so forth, and in vitro and in vivo methods to try to measure these off-target mutations. What I'd like to do in this talk is to tell you about a few new methods and papers that describe doing this comprehensively across the entire genome and doing it with new methods that give essentially a better magnifying glass, as you see it here in the picture on the right, to see better what the outcomes of these genome editing experiments are. So I'd like to just very briefly touch on four papers, and with the limited time, I can't go in detail. It's basically meant to give you a teaser, and if you're interested, you can read those papers in more detail where this has been described. So there are a number of examples over the last couple of years where this has been investigated, and specifically using new sequencing techniques. So this is a paper a couple of years ago in Nature Biotechnology. A repair of double-strand breaks induced by CRISPR-Cas9 leads to large deletions and complex rearrangements. So this paper is looking at the on-target undesirable effects. And you can see on the right side here in this graph, there are two stacked images. The first one on the left is looking around the target region, which is donated by the black lines, in about 5 or 6 kilobase scale. So this is looking much further around that region, and the stack on the right is looking at 100 base pairs. And so you can see that when you only look at very closely, at the target region, it might not look so bad, and there may be a little bit of a depression, but things look reasonably okay. However, when you look at it further out around this particular region, you see that there are pretty large depressions of read coverage and breaks in the read coverage. And so on the left side, on the bottom left, are just a couple of the undesirable effects that these researchers recognized, where portions from a completely different gene in the top were introduced at the break site, and then the second and third are inversions, either small or large, that were introduced by the CRISPR-Cas9. And so looking at this region with longer sequence reads and sequencing that full length gives you a better magnifying glass of what actually happened. And so just one more figure from this paper, if everything worked well, you would just see a dot, and the lines and the colorings indicate that there's quite a variety of different effects that can happen, both deletions in black and insertions in orange. And so these can now be measured and quantified. And then obviously that is a foundation to then adjust the experimental parameters to minimize or fix and avoid altogether these types of undesirable effects. Similarly, a newer paper from this year from a Chinese research group also looked at Cas9 cleavage products and also find large fragment deletions, very similar with these types of plots. Again, the coverage plot in orange on the right, you have the extent of the deletions, and you see again on the scale bar on the bottom right, 6 kilobases. So you really need to look at it at the kilobase scale, which now can be done, for example, with PacBio long-read hi-fi sequencing, to really comprehensively and accurately identify what happened at the site that was targeted. So again, this is an on-target method. This was a much earlier paper, really pioneering in Matt Porteous' lab from Stanford and Ayal Hendel in Cell Reports, characterizing and describing smart sequencing in particular as a great way to quantify genome editing outcomes. They described the system to work for all the common nuclease platforms, so not just CRISPR-Cas9, but also zinc fingers and talons and so forth. It can detect large insertion deletion gene editing events because of the long read lengths from smart sequencing. It's broadly applicable to any cell type and adaptable to any target site. And again, it allows then for optimization of these parameters to minimize, and it's highly accurate, fast, and inexpensive. On the right, what you see in the cyan color are the different off-target effects. And again, these can be fairly large pieces of DNA that are being inserted during the repair process of the double-strand break. And then the last paper that I would like to mention, and this is a new paper, it's a preprint by Ida Hoyer and Adam Amurs, the senior author. And I believe Adam is speaking at this very conference, so I would imagine that he's going to discuss this paper in much more detail. But in this paper, looking again at unforeseen CRISPR-Cas9 off-target activity, they not only look at the genome-wide by sequencing the entire genome in a reference-free, unbiased fashion, but also describe that the difference in the sequence of the two different alleles, the maternal allele and the paternal allele, have to be taken into account when you want to look at off-target activities, because the similarity of the sequence relative to the guide RNA will be different in some cases, depending on whether you're looking at the maternal allele and the paternal allele. And they show, this figure shows that you can have very different activities on the two different alleles. So more from Adam, I would imagine, in this conference, and it's a really, really terrific preprint that and very recent work that I would encourage you to take a look at. So in summary, for the first part of this talk, it is important to measure the outcomes of the genome editing comprehensively, both with regard to on-target and off-target effects, because for an efficient and safe procedures, you need to minimize and eliminate those types of effects. And there are new solutions available based on long-read sequencing that can measure these types of outcomes at the multi-kilobase scale around the target size, or, as I mentioned in the last example, for unbiased genome-wide sequencing, you avoid bias by the reference genome. You don't have to map reads anymore. You can do de novo assemblies. You avoid allelic effects and so forth. And so I think that's really the future of looking at these types of experiments and the outcomes of CRISPR-Cas9 experiments in an unbiased and comprehensive manner. And just for one related note, this month's cover of the journal Genome Research is an artwork by Arang Ri, who's at the NIH, a co-author on this paper that's shown on the right, about using high-fidelity long reads now to assemble and characterize the human genome much more comprehensively than before, even resolving centromeres and so forth. And so this is now possible, and it is efficient and it's cost effective to no longer rely on short-read sequencing and mapping to the reference genome, but you can get a much more comprehensive view of the entire, in this particular case, human genome or your genome of interest, to make sure that you're not having undesired off-target effects or on-target effects from CRISPR-Cas9 genome editing experiments. So in the second part of my talk, I'd like to switch and talk about opportunities that can leverage the really tremendous specificity of the Cas9 enzyme for targeted sequencing, and in particular regions that are very difficult previously to get any other way. And these are medically relevant. For example, in the neurobiology space, there are over 40 known inherited neurological disorders that are caused by these so-called repeat expansions. So these are simple DNA sequences that can be expanded in their repeated over and over from under 1,000 bases up to hundreds of kilobases. And the length of those repeated expansions determine the phenotype and whether you have a normal phenotype versus a disease phenotype. And the problem is that these are, for example, in the figure here, you have some triplet repeats or pentanucleotide repeats that because they are very low complexity or have an extreme sequence composition, they typically cannot be amplified efficiently or at all with PCR. So you can't, amplify them out from the genome, the patient and the person you want to test because of the nature of these particular sequences. And so you have to find alternative ways to try to target and enrich for this region. And the Cas9 enzyme provides really exciting opportunities there. So the way this can be done with a Cas9-based enrichment for targeted sequencing is that you have your DNA of interest on the upper left, and the first thing you want to do is block the 5' and 3' ends of the extracted DNA. You can do that simply by dephosphorylation because there will be a ligation step in the future, and you want to avoid ligating all of the DNA fragments. And so we're going to try to enrich for the one squiggle there in the middle that has the black segment, which is the target region of interest. And so then you design guide RNAs, 2 guide RNAs that flank this particular region. And again, as you'll see with in conjunction with long-read sequencing, you can design something that has, let's say 4 or 6 kilobases around the target region. So you only need 2 guide RNAs, and that piece of DNA will be enriched and excised in its entirety. And so there's no assembly or no tiling required and so forth. And so then you're taking advantage of the specificity of the guide RNA to then make 2 cuts and make a piece of DNA that incorporates that contains the region of interest, and significantly also does not have any of the blocked ends. So that means it is available for ligation. And so in conjunction with, for example, smart sequencing, you can then ligate these hairpin adapters. They can have barcodes which are here designated in purple, which I'll talk about in a minute, that allows for a high level of multiplexing, which makes it efficient. And so you can see that there was no PCR at all in this particular procedure, and that's why I'll refer to it as the no-amp or no amplification method. And now you have enriched for just the DNA that's ready for sequencing that contains the region of interest. So there are a number of examples here. This is a busy table, but basically this method allows for base level resolution of the expanded genomic region, an accurate repeat count, identification of interruption sequences. I'll talk about that in a minute. I mentioned it's free of PCR, so you don't have any PCR bias or copying errors. It's looking at the native DNA as it is, as it was extracted from the cells, and you can look at somatic mosaicism. So, and it's amenable to multiple kilobases, looking at long expansions. So I mentioned barcodes, and so here's just one example of an experiment where you can multiplex both several targets as well as several samples. So in this particular example, we mixed together 12 samples and 8 targets. So in the figure on the right, you see the different targets: FMR1 for Fragile X syndrome, HTT for Huntington's, C9orf72 for ALS, ataxin 10 for ataxia, and HBA and HBB for thalassemia, and the last two are CYP genes for pharmacogenetics. And all of those targets can be interrogated in a single experiment at the same time. That's all one assay. And of course, with the selection of the guide RNAs, you can customize what genes do you want to look at for this particular sample, and can have really high flexibility in customizing the targets that you would like to interrogate. And then the colored bars represent the different samples. So these are control samples that you see on the bottom, NA with a number from the 1000 Genomes Project, etc. And there are 12 samples, and all of them, again, are pooled in a single reaction, which makes this method really high throughput and cost efficient. And on the left there, you see some stats about how many total reads and reads per sample, reads per target. But on the y-axis, on the right graph, you can see that all of the bars are well over 50, and 50 is even overkill for generating a high consensus on both alleles. So all of these will give high confidence calls for the repeat expansions. We have developed, in addition, some useful visualization tools. So you can here count and see the expanded repeat. So for 3 different examples, for the Huntington's, the first 2, and then Fragile X, you see the normal allele and then the expanded allele. On the x-axis is the repeat count unit. And this, you see that the peaks are broadening for the expanded allele, and this is well known in terms of the biology, that because you have sloppy replication, that in the organism, in the individual, the exact number of reads can vary, and you get a good sense of how broad that distribution is. Another way to look at this is in what we call the waterfall plots. So here you see the 2 alleles and the reads belonging to each allele stacked up. You see with the colored, so the gray is CGG in this particular case on the left, which is the main repeat, but in orange you see AGG, and so you can immediately see that the normal allele had 2 AGG interruptions because it has 2 orange lines on the top left, and then the expanded allele has only 1, and that's significant for the risk assessment of how likely it will be for a mother to give birth to a son that would have the disease and so forth. And on the right for Huntington's, you see a more complex picture with the main CAG repeat in gray, and then a number of different sequence triplets as interruptions, and that all can be quantified and seen really clearly. And then, the typical IGV visualization. So at the sequence level, you see how clean the hi-fi reads are, and then you can zoom in and look at the sequence context and see really with high clarity. You can see there are certain deletions that correspond to the different lengths of the CAG repeats. You can look at the specificity of the guide RNAs very easily across the entire human genome. So in this particular graph, you see the human genome. You see the numbers on top of the graph, the chromosomes 1, 2, 3, all the way to X and Y, and then the reads. Because they're kilobases long, you can map them uniquely where they came from in the human genome. And in this particular experiment, 4 targets were 4 loci were targeted: Huntington's, ALS, SCA10, and FMR1. And you can see there's very good read coverage. And then by the errors donate 2 instances in the human genome where there was also read coverage, meaning there was some enrichment. And you can then look at those and find that this was caused by sequence similarity of those particular regions on chromosome 2 and chromosome 9, having some similarity with one of the 4 guide RNA pairs. And you can look at the sequence at the base level. You can quantify the off-target effect based on the height of the peaks. And so it gives you a really good, comprehensive and unbiased view of whether you need to optimize things further. Now, for the targeted sequencing, that's fine because you can just remove those. But again, coming back to the first part, for genome editing experiments, you would look at that and say, well, maybe I need to optimize my protocols and the guide RNAs further in order to avoid potential off-target effects. So just to summarize, this method, the no-amp targeted sequencing method, allows for a high level of multiplexing, currently up to 48 barcoded adapters for sample multiplexing. And for targeted multiplexing, we have 18 validated guide RNAs available, but as I mentioned, it's expandable and customizable to other regions of interest, and you can mix and match to your area of research. There are a number of publications already available, so I just wanted to include that here, and we'll make the slides available so you can click and learn more about what the research community has already achieved with this method, and we certainly see a lot of opportunities for the future. So the summary of this part 2 is that this amplification-free targeted enrichment, leveraging the CRISPR-Cas9 enzyme and specificity, can be used to target hard-to-amplify regions. You have no PCR, bias, or errors. You can multiplex targets and samples. In conjunction with smart sequencing, you span the full region of interest with high base accuracy and uniform coverage, and you can characterize mosaicism. And for the particular example, not limited to that, of course, but just as one example for repeat expansion disorders, it allows for accurate repeat counting for both alleles, identification of interruption sequences, and the characterization of mosaicism. So with that, I hope I've given you an overview of these two topics. And as always, I understand that questions can be emailed, which I'll be happy to answer. And so again, thank you for the organizers, and thank you for your attention.
Okay, great job. Well, we're all set pretty much from here. I'll just do some quick editing on the front and back ends and get your presentation ready for next week.
Wonderful. Thanks so much again. Nice to talk to you again.
Awesome. Yeah, you as well. If anything comes up, feel free to reach out. But if not, we will go from here.
Okay, great. Thanks so much. Have a good day.
Thank you. You too. Bye-bye.
Bye.